bacteriophage. T4 is a bacteriophage that infects E. coli bacteria. The phage is a member of T-even phages, a group which includes enterobacteriophages T2 and T6. It's one of the largest phages, encoding roughly 200 genes, and it was the first prokaryotic organism providing evidence of gene splicing through presence of introns in the genome. T4 is capable of undergoing only a lytic life cycle, and it does not undergo lysogenic life cycle. T4 has been extensively studied, and it has a history which is very rich in advancement of genetics. Some of the essential ideas of genetics came from the study of T4, which includes basics of genetic code, ribosomes, mRNA, and codon studies. It's surprisingly the science that has learned so much using a virus which is extremely very complex. Nonetheless, we owe our much knowledge to the current T4 scenario. Was the genome and structure of T4 bacteriophage. The elongated icosahedral head of T4 phage contains linear double-stranded DNA genome, which is about 169 kbp long and encodes 289 proteins, having two accessory proteins, that is HOC and SOC. HOC is highly antigenic outer capsid protein and SOC is small outer capsid protein. Binding of SOC stabilizes the capsid and resists osmotic shocks. The T4 genome is terminally redundant, and it first replicated as a unit, then several genomic units are recombined end-to-end to form a concatemer. When packaged, the concatemer is cut at unspecific positions of the same length, leading to several genomes that represent circular permutations of the original one. The genome is AT-rich and contains modified bases in the form of 5-hydroxyl methyl cytosine rather than cytosine, which protects the phage DNA from many host restriction systems and from phage encoded nucleases that can degrade cytosine containing host DNA during infection. The T4 genome bears eukaryote-like intron sequences. The tail fibers of T4 pins and base plates are involved in binding to the lipopolysaccharide receptor of the E. coli. If these fibers are going to bind to the E. coli, they need a receptor site. Without the receptor site, no virus can bind to the cell. The T4's tail is hollow so that it can pass its nucleic acid into the cell. The tail attaches to the host with the help of tail fibers. What is the life cycle of T4? As we earlier said that T4 has only a lytic life cycle and not a lysogenic life cycle. So all the steps that are going to be followed are of lytic life cycle. The first phase is T4 absorption and injection. Phage binds to the lipopolysaccharide and tryptophan receptor on the bacterium. T4 looks from the susceptible bacterium with tail fibers. The tail fibers recognize the membrane first. Then when they are bound to the cell, the base plate goes under conformational changes which causes the sheath to contract, piercing the cell membrane, which allows the viral cold to enter the cell and release the double-stranded DNA viral genome. The outer sheath contracts, driving the internal tail tube into the cell. The gene product facilitates digestion of the peptidoglycan layer of the bacterium to reach the inner membrane. The gene product here is GP5. The tail membrane electrochemical potential is required for transfer of T4 DNA to cytoplasm. The empty capsid remains extracellular in the meanwhile. Second is replication and expression of gene. Once T4 DNA is in the cytoplasm, it specifies a highly organized and coordinated program of gene expression with the help of three promoters. One is PE, that is early gene, second is PM, that is middle genes, and third is PL, that is late genes. Some of the first genes which are transcribed are called as immediate early encode enzymes which break down host DNA. When the host DNA is broken down, the host nucleotides produce viral DNA. It encodes the proteins needed for DNA synthesis, which leads to building up of capsid and tail structures. The host enzyme RNA polymerase transcribes and translates the expression of early genes. Delayed early and middle genes encode 20 proteins that are all involved in viral replication. There are two distinct replicative processes of T4. First is replication occurs in bidirectional manner with multiple origins of replication within the genome. The replication can start from any side. The first several rounds of replication are initiated by RNA primers, which are synthesized by host RNA polymerase. These RNA primers can travel to their complementary region in double-stranded DNA and displace the other strand, which produces a structure that is called as R loop. The attached RNA can now act as a primer for the leading strand of DNA replication. The lagging strand is then synthesized using the replicative helicase GP41. DNA replication is finished by GP30, which is T4 encoded ligase as well as the host DNA ligase. After several minutes of infection, host RNA primers cannot be used because the promoting recognition specificity is altered. Second stage. At this point, the virus uses its own replicative machinery to transcribe its late genes. This process is known as GP45 sliding clamp model. During this, early gene products alter the host RNA polymerase in two ways for expression of middle genes. It modifies RNA polymerase and does the translation. The enzyme is thereby all to read through a transcription terminatal by anti-termination mechanism. This was for middle genes. For late genes, transcription is coupled to replication. Promoters for these genes again differ from sigma 70 promoters and require an alternative sigma factor which is encoded by T4 regulatory gene. Then assembly and release. We all know after penetration, attachment and then uncoating and replication we get assembly and release. Once late genes are expressed, the viral base plate is first assembled and then it attaches to tail and tail fiber proteins. These three different protein pathways combine to form a mature T4 phase capsid. The DNA is packaged into a mature capsid protein by packing and cutting the concotamers using a terminase complex found at the end of concotamerized DNA strand. The terminase complex binds to the capsid head and moves DNA into the empty capsid. The capsid also encases necessary enzymes for future infections like as virally encoded DNA polymerase. The viral encoded enzyme holin creates the holes in the inner membrane of the bacterial host cell, which allows lysozymes to exit, and it degrades the peptidoglycan cell wall. Cell lysis subsequently follows, releasing a shower of bacteriophage progeny into the extracellular space. These are the questions you have to answer before finishing it.